Look at your hand right now. Seriously, just take a second and really consider what it's actually made of. We usually just think, skin and bone, right? But underneath all that, there are literally trillions of tiny living chambers. And they're actively working, communicating, and replicating at this very second, just to keep you alive. So what are you fundamentally made of? It's a question that completely pulls us out of our everyday macroscopic lives and drops us straight into a microscopic world that is honestly as complex and dramatic as any sci-fi movie.
Welcome to today's Explainer. I am absolutely thrilled to have you here. Today, we are going to unpack the architecture of life itself. We're taking a serious, straight to the point, but still really fun journey down the ladder of biological complexity. Let's dive right in.
Okay, here's our roadmap for today. 1. The blueprint of life. 2. Eukaryotes, cellular cities. 3. Prokaryotes, bacterial survivalists. 4. Viruses, the acellular hijackers. And finally, 5. The viral life cycle.
Kicking things off with Section 1, the blueprint of life. Let's cover the fundamentals of cytology.
To really grasp the grand architecture of life, we have to start with the foundational bricks, the cell. It's the basic structural and functional unit forming absolutely all the tissues in an organism. Now, the word itself comes from the Latin cellula, which translates to small chamber, and honestly, that's a perfect way to visualize it. It's a self-contained little room where the actual business of life gets done. Each cell is its own living entity, completely capable of autonomous reproduction. And the scientific study of these units, that's called cytology, which comes from the Greek word for cell. But cytology isn't just some abstract textbook science. It's a critical, everyday tool in medicine. By analyzing these tiny chambers at the microscopic or molecular level, doctors can diagnose everything from a severe infection to developing cancers.
Now, biology draws a really hard line in the sand between the two fundamental types of cells. You've got prokaryotes and eukaryotes. Prokaryotes, well, they're the minimalists. They have absolutely no true nucleus. Their DNA just kind of floats freely in the cytoplasm. They lack any membrane-bound organelles, and they are always unicellular. A great example is the E. coli living in your gut. Eukaryotes, on the other hand, are the complex heavyweights. They feature a true protected nucleus to house their DNA, plus a highly complex internal organization that is just packed with organelles. And they can be unicellular, like yeast, or massively multicellular, like us. Moving on to Section 2, Eukaryotes, cellular cities. We are going inside the microscopic metropolis. I want you to think of a eukaryotic cell not just as some random blob of jelly, but as a bustling, highly organized city. Every internal structure, which we call an organelle, has a super specialized job. The nucleus, that's the city's highly secured data center. It stores and tightly protects the vital DNA blueprints. Then you have the mitochondria. These are the powerhouses, just furiously converting glucose into usable cellular energy called ATP. The endoplasmic reticulum and the Golgi apparatus. They team up to act as the city's factory and postal service, constantly synthesizing, packaging, and shipping out proteins and lipids. And of course, you can't run a city without waste management, right? That's exactly where lysosomes come in. They act as the recycling centers, using potent digestive enzymes to break down and recycle cellular trash. But hey, the city infrastructure certainly doesn't stop there. The sheer level of complexity required to sustain multicellular life is just staggering. For instance, if you're a plant, you have chloroplasts acting as massive solar panels for photosynthesis. Cells also rely on peroxisomes, which are basically chemical detox centers that neutralize dangerous byproducts like hydrogen peroxide. Then there are vacuoles. These are massive storage tanks holding water and nutrients to keep the cell balanced. And literally connecting all of this together is the cytoskeleton, a sprawling structural protein highway that gives the cell its shape and manages internal traffic. Finally, guarding the entire metropolis is the plasma membrane. Think of it as the strict phospholipid border control that dictates exactly what is allowed in and out. It's this massive, intricate coordination that allows complex life forms, from towering trees to humans, to even exist in the first place. Let's step down the complexity ladder for Section 3, Prokaryotes. Bacterial survivalists, the rugged, simple, and ancient ones. So, if eukaryotes are sprawling modern metropolises, prokaryotes are the rugged, ancient survivalists. Sure, they lack a fancy nucleus and complex organelles, but whatever you do, do not underestimate them. Prokaryotes, like bacteria, are equipped with a highly specialized arsenal designed for one thing: survival. They often deploy a capsule, which is this sticky polysaccharide shield that helps them literally hide right under the nose of our immune system. Beneath that is the cell wall. This is rigid peptidoglycan armor that protects them from bursting and defines their shape, whether they are spherical cocci or rod-shaped bacilli. To interact with their environment, they use fimbria and pili. Think of these as hair-like grappling hooks that let them stick to our tissues or even swap DNA with other bacteria. And for getting around, they use flagella, which function exactly like microscopic outboard motors to propel them through fluids. So their simplicity really is their strength. But their true superpowers go way beyond that. Many bacteria carry around plasmids. These are small circular fragments of DNA entirely separate from their main chromosome. And get this, these plasmids often contain the exact genes needed to neutralize the very antibiotics we use to fight them. It's a real game changer. And what happens if the environment becomes totally lethal, like zero nutrients or flooded with deadly chemicals? Well, some bacteria pull off their ultimate survival trick, sporulation. They basically pack up their essential DNA, form an incredibly tough outer shell, and go into deep hibernation as a spore. They just wait out the hostile conditions until it's safe to bloom again. They are literally the ultimate biological preppers. Which brings us to Section 4, viruses, the acellular hijackers. This is where biology completely breaks the rules. I want you to let that question sink in for just a second. We've spent all this time marveling at the incredible autonomy of the cell. But what if a devastating, globally disruptive infectious agent isn't even a cell at all? It has zero metabolism. It doesn't consume energy. Left entirely on its own, it is completely inert. By definition, a virus is a very small, acellular, infectious agent. That means not a cell. It contains only a tiny strip of instructions, either DNA or RNA, and that's wrapped tightly inside a protective protein coat called a capsid. That is the whole shebang. When it's fully mature and just waiting outside a cell, we call it a virion. And here is its most insidious trait. Because it lacks all the normal machinery of life, it absolutely requires a host cell to multiply. It is a biological hijacker, plain and simple, that has to infiltrate our cellular cities to survive. And man, viruses come in some strikingly mechanical architectures. In fact, the shape of that protein capsid is a major way we classify them. First, you've got helical symmetry. This is where the proteins arrange in a flexible, tubular, or cylindrical shape around the genome. The tobacco mosaic virus is a classic example. Then we have icosahedral symmetry. These are spherical, incredibly stable structures, built almost like 20-faced geodesic domes. It's brilliant because it maximizes structural protection while using minimal genetic code. And finally, my personal favorite, the complex structure. These combine different shapes and often feature things like tail fibers and base plates. If you've ever seen illustrations of bacteriophages, the viruses that hunt bacteria, they use this complex structure. Honestly, they look and function exactly like microscopic alien landers dropping down onto the surface of a cell. We also classify viruses by the armor they wear. Enveloped viruses literally steal a lipid envelope from their previous host's cell membrane right as they exit. This stolen disguise is super effective. It helps them fuse easily with new cells and partially evade our immune system. HIV and SARS-CoV-2 are prime examples of this. Non-enveloped viruses, on the other hand, don't even bother with a stolen membrane. They rely entirely on their naked protein capsid. And actually, because they don't have that delicate lipid layer that gets easily destroyed by things like soap or alcohol, these naked viruses are notoriously tough. They are environmental survivors capable of lingering on surfaces for a shockingly long time. Poliovirus and HPV are great examples of these tough, naked capsids. Now for our final chapter, Section 5, the viral life cycle, the hostile takeover. So how do these tiny, inert packages of rogue DNA actually manage to conquer the massive, complex cellular metropolises we talked about earlier? Well, it plays out in a terrifyingly efficient sequence known as the lytic cycle. Step 1 is attachment. The virus acts exactly like a specific key fitting into a very specific lock, binding perfectly to receptor proteins on the host cell's surface. Step 2 is injection. The virus breaches the perimeter and shoots its DNA or RNA straight into the cell. Step 3 is the hijacking. The virus injects unique proteins that forcefully shut down the cell's normal day-to-day functions. It essentially unplugs the factory, stops it from making what your body needs, and forces it to expend absolutely all of its energy replicating the viral genome and assembling new viral parts. That healthy factory instantly becomes a massive viral reproduction plant. Step 4 is release. Once the cell is filled to the absolute brim with newly assembled viruses, they violently burst through the cell membrane in a process called lytic release, which leads us inevitably to step 5, host death. The original cellular city is completely destroyed, leaving a gaping hole in its membrane, while hundreds or thousands of new viruses spread out to find their next target.
But here's the thing. The lytic cycle isn't even the only threat. Bursting the cell isn't the ultimate hijacking. Certain viruses, like human papillomavirus or HPV, they don't just kill the cell. They actually alter the DNA of their host. They stitch their own rogue genetic code right into the host's blueprints. This can activate cellular oncogenes and inhibit tumor suppressor genes. In doing so, these tiny, completely non-living entities can rewrite the fundamental instructions of a complex eukaryote and actively promote cancer development. It really makes you pause, doesn't it? Just pondering the incredibly fragile balance of microscopic power happening inside of us right at this very moment. Thank you so much for joining me on this explainer. Keep learning, keep questioning, and I'll catch you next time.